Trisomy. Trisomy is the type of the polysomy in which we see three chromosomes of a particular chromosome. There are three copies of a particular chromosome instead of the two copies if the individual is diploid. Trisomy is of two types. It can be autosomal trisomy, it can be sex chromosome trisomy. In case of autosomal trisomy, the important type of the syndrome is Down syndrome. And in case of sex trisomy, Klinefelter syndrome is the type of trisomy. There are different types of the trisomy. The first type is primary trisomy. In case of primary trisomy, there is one complete extra chromosome. If one complete chromosome is extra, then it is called as primary trisomy. While in case of partial trisomy, if a small fragment of the DNA or small fragment of the chromosome is present, then such a trisomy is called as partial trisomy. While in case of secondary trisomy, that is extra chromosome has duplicated arms. The fourth type of the trisomy is called as tertiary trisomy. Tertiary trisomy is the type of the trisomy in which the arms they are copied from two different chromosomes. Common trisomies which have been seen in case of the humans is trisomy of 21, chromosome 21, trisomy of chromosome 18, trisomy of chromosome 13. These are the three trisomies which are of autosomes. While in case of sex chromosomes, the trisomy has been seen in the form of Klinefelter syndrome. Trisomy 21 in the humans is called as trisomy 21. There is approximately one birth out of 700 births which has this kind of trisomy, and this disease or this syndrome is associated with the maternal age. In this diagram, we can see that how a primary oocyte, if there is non-disjunction, is converted into the trisomy. If there is non-disjunction that we can see here, it is ultimately converted into 3 chromosomes instead of 2 chromosomes. Here we can see that there are 3 chromosomes. But if there is normal meiosis, then there will be 2 chromosomes. If there are 2 chromosomes, then it will be a normal cell, while if there are 3 chromosomes seen of a particular chromosome, then that process is called as trisomy. Trisomy can be formed due to non-disjunction, either in case of first meiotic division, like here. In this case, there is first meiotic division and it is non-disjunction. As a result of non-disjunction, we can see that the trisomic cells will be produced. While trisomic cells can also be produced if there is non-disjunction during the second phase of the meiosis. If here is non-disjunction, then we can also see that some of the cells which will be produced, they will be trisomic in nature. This is the diagram of the trisomy 18. And the name of this syndrome is Edwards syndrome. In this syndrome, we can see that all the set of the chromosomes, they are in diploid form. There are 2 chromosomes. These are the 2 chromosomes. These are the 2 chromosomes. They are homologous to each other. But in case of chromosome 18, we can see that there are 3 chromosomes. So in case of chromosome 18, because there are 3 chromosomes, we will call it trisomy of chromosome 18 or trisomy 18. The disease which is caused due to trisomy 18 is called as Edwards syndrome. This is the second example of the trisomy 13. In case of trisomy 13, we can see that all the chromosomes are in diploid form or in homologous form instead of chromosome 13, where we can see that there are 3 chromosomes. So trisomy is the type of the polysomy in which there would be 3 chromosomes of 3 copies of a particular chromosome.